So for this video, I'm going to be talking about regulation of the complement system. The complement system is an essential element of the innate immune response that becomes activated upon recognition of molecular patterns associated with microorganisms, abnormal host cells, and modified molecules in the extracellular environment. So these are outside of the cell. And it's active in both innate and adaptive immunity, but it really focuses on the innate immunity. Through these activities, complement function in the first line of defense against pathogens, but also contributes significantly to the maintenance of homeostasis and prevention of autoimmunity. And then activation of complement and the subsequent of biological responses occurs primarily in the extracellular environment. So again, it occurs primarily outside of the cell. And then major effect within the innate immunity. So like I said, it's in both innate and adaptive, but mostly in innate. As a universally distributed defense mechanism in blood and interstitial fluids, complement is one of the first lines of defense against pathogenic microorganisms that breach or get past the mechanical and chemical barriers of our body. So when they get past those barriers like the skin or past chemical barriers like mucus and stuff like that. It is a germline encoded system, which is the genetic material passed down in our body. So any material that is passed down of more than 50 circulating and membrane-bound proteins. And then complement was first identified as the heat-sensitive fraction of human plasma that complemented antibodies in their ability to kill bacteria. So that's where they get the name complement. Complement activation. So before you can have regulation of the complements, you have to have activation. And upon complement activation, structural rearrangements, cleavages, and the assembly of proteolytic and lytic complexes occur. In this way, complement can be ubiquitously present in an inactive form, but become active locally. So complement is activated through the classical pathway, which is CP, the lectin or lictin pathway, LP, and the alternative pathway, or AP. The recognition of invading microorganisms by the complement system can occur directly via recognition of pathogen-associated molecular patterns, or PAMPs, PAMPS, by soluble pattern recognition molecules, or PRMS. And then after activation, you have regulation. Regulators of complement activation, even healthy host cells by the classical pathway or lectin pathway, PRMs may become tagged by C3b due to the generation of C3 from the AP pathway, or through the bystander effect, where the C3b generated in the vicinity becomes attached to the host cell. One of the most potent and best studied complement regulators is complement factor H, or better known as FH. FH is a very strong negative regulator of complement on host surfaces, to which FH binds through excuse me, sialic acid, and then by binding to host-specific glycans, FH is able to distinguish between self and non-self, and then that prevents complement activation on host cells. So by binding to the host glycans, FH is able to distinguish which is part of the body and what's not. On microorganisms lacking these protective patterns, FH is absent and complement activation is then allowed to proceed. And then complement activation during normal homeostasis The central component of the complement system is C3. Like I stated before, the activation of the three pathways, CP, LP, and AP, results in cleavage of inactive C3 protein into the functional fragments C3A and C3B. So it's broken down into those two fragments. C3A is an inflammation mediator, and C3B is an opsonine. I'm not really sure how to pronounce that. Which can bind covalently and tag any surface in the immediate proximity to the site of its generation. And the complement system has the potential to be extremely damaging to host tissue. That means its activation must be extremely regulated. So it's important that activation of the complement system is regulated. It's regulated by complement control proteins, which are present at higher concentrations than they are at lower concentrations in the blood plasma than the complement proteins themselves. So some complement control proteins are present on the membranes of cell preventing them from being targeted by complement. One example of that is CD59. That's also known as protectin, which inhibits C9 during the formation of the membrane attack complex, and that is formed on the surface of pathogenic bacterial cells as a result of the activation of the host three pathways. Again, those are alternative, classical, or lectin pathways. Excuse me. Classical pathway is inhibited by C1 inhibitor, which binds to C1 to prevent its activation. So this picture right here has all three pathways. We have the classical pathway, binding of C1q to antibody antigen complex CRP, and then we have the lectin pathway, binding of MBL or phycolins or to pathogen surfaces. And then you have the alternative pathway, spontaneous breakdown of C3 to C3H2O. And it just goes through the processes of both, starting with the classical and then the alternative. And then as you can see that this one has a lot more going on with it, but then they all end up down here. It's a membrane attack complex.